So last class what did we discuss? We were discussing about the various genetic transformations in plant cells. What are the different ways in which plant cells can be transformed? So we were talking about that generally in literature you will find that agrobacterium-mediated transformations are used. Agrobacteriums themselves are natural engineers to they have the machinery to successfully transform the plant cells through a section of DNA present in them, plasmid DNA, which is called as tDNA, which gets stably integrated. So how we have exploited is that tDNA, inherent tDNA present in the agrobacterium, that is disarmed, which is taken away, and another vectors which have this segment of tDNA, which agrobacterium has the machinery to integrate this plasmid vector, which has been exogenously added into the agrobacterium to integrate into the plant cells. So now what are the different kinds of vectors which we were discussing last time? The most commonly used vectors are binary vectors. Now binary vectors, why are they called binary? Origin of replications. They can replicate in E. coli as well as in agrobacterium. And then what else? The conjugal transfer can take place between E. coli to agrobacterium and from agrobacterium to plant cells. Now I was also talking about other vectors which can be used for genetic transformation in plant cells. These are intermediate vectors. What are these? Like for example PBR322, you will be hearing if you work in this area. These are smaller size vectors in E. coli which contain the tDNA, and they are used to overcome the problems because of the large size of the Ti plasmid. So as I was saying, there is a helper plasmid present in the agrobacterium which is disarmed. Now this is called as helper plasmid, which will have virulence regions. Now this small plasmid, it can multiply in the E. coli and then through conjugal transfers it can be brought in the agrobacterium. Now these plasmids in agrobacterium, they cannot multiply, but then they can be transformed, they can be used to transform the plant cells. Now co-integrative, sometimes what happens that because of there is homologous recombination. Now in the agrobacterium, some of the tDNA regions which have been disarmed, but the other regions, if there is a homologous if there is a similarity between that DNA component to the DNA which is present in these vectors, then transformation would take place, recombination would take place, and there will be an exchange of genetic material, and this tDNA where your MCS region is there, this gets integrated into the entire one single plasmid, which can then act as a complete Ti plasmid for the transformation to happen in the plant cells. So then it is called as co-integrative plasmid. Then helper vectors. Helper vectors, either you have your inherent Ti plasmid which has been disarmed. Disarmed means the oncogenic regions have been, the tDNA has been taken out. So, or you have separate plasmids which help in the transformation event, which is they will have regions which are transfer regions which will help in the transport of this tDNA of your small integrative vectors. Now these are small plasmids maintained in E. coli. These are helper plasmids for E. coli to agrobacterium transfer. They contain transfer regions and mobilization regions which allow the transfer of the conjugation deficient intermediate vectors into agro. Suppose if there is conjugation which is absent, not all vectors are conjugal vectors. So if conjugation is absent, then you need helper plasmid which can help in the transfer of these vectors into the agrobacterium. So this is what I was saying, co-integrative vectors where homologous recombination, entire one co-integrative plasmid inside the agrobacterium can be obtained. Now what are the different transformation techniques? Either what is well known which is being used in laboratory is co-culture technique. One is that, and the other is direct techniques in planta transformation. Direct, the plant is transformed. Now co-culture with tissue explants means it begins with in vitro cultures, and from the in vitro cultures regeneration is done to get the transformed plant. Otherwise directly the plant parts can themselves will get transformed, either the seed itself or the zygote, or at the time of floral pollination, just before pollination when the flowers come out, there is a flower dip method or floral dip method through which Agrobacterium can get into the plant, and after pollination can transform the zygote, thereby in the progeny you will see transformation happening. Then direct gene transfer or physical delivery methods. So, direct gene transfer, either we are using Agrobacterium as a tool to transfer, or in direct gene transfer methods, for example gene gun, which I think you must have heard about, and there are other methods which we will see. So, Agrobacterium-mediated methods, what is done is, the prerequisites for Agrobacterium-mediated gene transfer, it involves the explant must produce acetosyringone. What was acetosyringone? So, this was used to induce the virulence genes present in the Agrobacterium. So, acetosyringone, so the type of explant, the age of the explant also sometimes matters for the successful transformations. So, there has to be production of these signaling molecules which can attract the Agrobacterium and increase the proximity between the Agrobacterium and the plant cells. Now, the explant must produce acetosyringone or other active compounds, other phenolic compounds, in order to induce the virulence genes. Then the Agrobacterium should have access to cells that are competent for transformations. Sometimes it is observed, although transformation is done, but regeneration does not happen. For regeneration to happen, you need competent cells for transformation and then totipotency to be maintained in the transformed cells so as to reach into the regenerant plant. So, which is a difficult task, not that easy. So, now for Agrobacterium successful transformation, in access of the competent cells is necessary. Not all cells will be competent in nature. Sometimes you will see that similar method, but you will keep on repeating, only some will be successful, the others may not, because of all these reasons. Not all cells in the callus will be of the same biochemical nature. Then explants used for transformation, the type of explant, the age of explant. Now, it is generally preferred that they should be actively dividing cells when transformation should be done. Why? Young explants are preferred, not old. Why do you think? Even for transformation, regeneration is the second step. For successful transformation to happen, it is preferred that you should work with younger explants with rapidly dividing cells if you are working with cell cultures. So, but let's do a comparison between matured cells in suspension and the secondary cell wall comes when organogenesis and it is in the tissue present, isn't it? There can be, the probability can be higher. What else? So, matured cells, is there one major difference between matured cells and immature cells? If you remember, I was telling in the earlier classes that the younger cells, the vacuoles present are small, the cytoplasm is more dense. So, the probability is higher. In mature cells, the vacuoles become large, the cytoplasm is less dense. So, denser is the cytoplasm, higher is the probability for transformation. So, and moreover, vacuoles contain enzymes which can rapidly degrade the extracellular DNA. So, it is preferred that you use younger cells in comparison to matured cells. In literature, you will find the cells which are used or the tissues used, they vary from callus, all kinds of in vitro cultures can be transformed. Callus, suspension cells, protoplast, then tissue, whole organ, entire plant. So, there is a wide range. Now, after explants have been co-cultured with Agrobacterium, how to detect that which cells are successfully transformed or not? We have already read now that based on the selection marker or the marker system present, whether it is histochemically based assays which can be used, or antibiotic selection medium or herbicide selection, medium can be used to increase the probability that you will end up in successfully transformed cells with subsequent subculturing. It, apart from the marker or the selective agent, Agrobacterium inhibiting growth inhibiting compounds, other antibiotics like carbinsulene, through Agrobacterium is sensitive towards it. So, is also used. Why do you think? And when is this used? Or when should it be used? For transformation, what are you doing? You are bringing the Agrobacterium in close contact with the cells, but ultimately what do you want? Transformed plant cells. You can't keep growing Agrobacterium because it will overgrow, which will subsequently lead to loss in the viability of the cells and the growth. So, after successful transformation, successive culture has to be taken away from the Agrobacterium, has to be removed. So, after 48 hours, generally 48 hours is the timeline for exposure with the Agrobacterium, and then it is kept on the high concentration of carbinsulene or any antibiotic where Agrobacterium is sensitive towards. So, then subsequently with reduced concentration of the antibiotic you keep subculturing, and after subsequent subcultures the antibiotic is removed from the medium, because antibiotic itself can be inhibitory to the plant cell growth, which you would not like to have for the transformed cells. In general, transformed cells, because you have genetically manipulated them, so their growth rates are compromised. So, the selection marker systems, the toxic level. So, what has to be optimized? The toxic level of the selection agent, it is very species. So, it is species specific. The time of exposure is species specific. The antibiotic concentration is species specific. So, that has to be optimized. So, this is the entire protocol, you can go through it. This is for your reference, the theoretical protocol, how it is done. So, what is done is you choose an appropriate explant, then you surface sterilize the explant. The explants are brought in, you grow the Agrobacterium in suspension for 24 hours. You look for the growth phase, because then the Agrobacterium cultures are active. So, generally log phase cultures are preferred. Once the Agrobacterium has grown, there are different strains of Agrobacterium which are present and which are virulent to infect the plant cells, but it is very highly species dependent. So, if one Agrobacterium, like for example Agrobacterium LBA 404 or 9402, so these are different strains which are known to be highly virulent, but it is not necessary that because it has worked with one plant species it will work equally with the other. So, you need to choose the Agrobacterium strain, grow it, active phase, log phase cells are used. Then even for exposing the cells you can use different techniques. What is needed? Either you create a wound in the explant and then expose the suspension to dip that explant, wounded explant into the suspension. Now, the time of exposure also varies. You cannot keep it for more than 15 or 10 minutes. So, generally 10 to 15 minutes you keep it exposed, then you need to wash it so that excess growth of Agrobacterium does not happen when you are keeping it in the for incubation for 48 hours. So, you can vary the time there of the exposure time. You can vary the Agrobacterium concentration, the cell suspension concentration. You can have different ODs when you are using the culture to co-cultivate with the plant cells. Then you can vary the type of explant, that can be varied. Then I was talking about the wounding. Now, wounding itself can be created through different ways. So, people have found that using a syringe filled up with the Agrobacterium suspension, you make injuries on the midrib of the leaf explants, and that has given rise to successful increased efficiency in transformation. Then people use simple dip method where they expose the explant wounded by a blade or by creating holes in the leaves or explants and then exposing it. So, there are different factors which need to be optimized. It looks easy, but there are number of factors which need to be optimized to get a successfully transformed culture. Then in plantar transformation, generally used is the floral dip method, in which the flower is dipped in the agrobacterium suspension and then kept for incubation for some time, such that it resides. It is said that either or the seeds themselves can be surface sterilized and dipped in the suspension, such that the agrobacterium can transform the seeds, and once they regenerate or when the seedling comes out, they infect the tissues, the aerial parts or the ground part regions. Or when the seeds, when the flowers are dipped with the agrobacterium or brought in contact with the agrobacterium just before pollination, during pollination or after pollination, the agrobacterium present inside may infect the zygote or may infect first hand the germ cells which are going to get involved in the zygote formation, thereby leading to progenies which are transformed. Now, talking about direct gene transfer methods, electroporation is a well known technique which is used even for bacterial transformations. Then what are the two different types of electroporations which are frequently used? These are low voltage long pulse methods, means time duration is increased but lower frequencies are used. High voltage short pulse method. Generally it has been found that high voltage short pulse methods are found to work better than the low voltage long pulse methods, because the damage to the DNA is lesser in that case. Now what is done? The optimal voltage. So what can be optimized? The voltage which you are using, the time for which it is being applied is optimized. Then transformation frequencies are increased several folds by adding certain other techniques like heat shock or adding your PEG. Why do you think it helps? Have you heard about competent cells? How are they made competent? Competent cells means they become more susceptible to uptake of the extracellular DNA. How are they made susceptible? Any divalent, even magnesium can be used. How is it making it susceptible? So thereby increasing the proximity between the extracellular DNA and reducing the repulsion, because both are negatively charged. So that is the way. And what about PEG? So what it does? So what is it actually doing? Everything is now facilitating easy uptake. Polyethylene glycol is also very frequently used. DMSO, polyethylene glycol, yeah.
It reduces surface tension and makes the membrane more permeable.
Then, so what is done is, in chemical methods, as we were talking about, the cells are first made competent. So divalent ions can be used to reduce the charge repulsion, such as to increase or increase the proximity between the two DNA. The other thing is that there is a usage of, people manipulate pH, people add polyethylene glycol in the medium, and the concentration of the DNA is also manipulated to enhance the efficiency of the transformation. Now some of these methods you need to go back and check. What they do is they will try to condense the DNA or concentrate the DNA, and concentrating the DNA, reducing the proximity between the extracellular DNA and the plant cell membrane would increase the probability of success. So that is what these things do. Now lipoinfection, they are also the extracellular uptake of the DNA can be improved by encapsulating them in the form of liposomes. Now liposome intake is a well known phenomena in cells. So liposomes, they not only will increase the uptake of the DNA, but they will also protect the DNA from the nucleases. So that way also it helps. Now what are liposomes? I hope you know. What are liposomes? They are similar, they will have both hydrophilic, hydrophobic ends. So, like phospholipid bilayer, and they will encapsulate the DNA within. Now they can be induced to fuse with the cells or the protoplast using PEG and can therefore be used for gene transfer. Then there are several advantages. What all different advantages with lipoinfection? Low cell toxicity, stability and storage of nucleic acid can be improved by encapsulation, then high degree of reproducibility and endocytosis is a well known phenomena already present in the cell, so that can be easily taken up by the cells. Now calcium phosphate precipitation, another method. Calcium chloride anyways is being used. If the phosphates are present, then it condenses the DNA, precipitates the DNA such that because it is condensed, it reduces or allows the cell bring it much closer to the cell membrane, thereby increasing the probability of uptake of the extracellular DNA. Microinjection, this is direct gene transfer in which a microinjection assembly is used. It is not that easy. Under the microscope, you hold the cell using or by freezing it onto a slide using polylysine. Polylysine, because of the charges present, it will try to stick it at one place, and then suction pressure is created such that it does not move, and then through that micro injection assembly, the DNA is tried to either directly put in through the cell membrane into the cytoplasm or into the nucleus of the plant cell. Now what other direct methods can be used is, in literature you will see that people have used fiber mediated DNA delivery, in which very small size nanofibers are used, silicon fibers are used with high speed so that they can impregnate the plasma membrane, the cell wall, and the DNA is delivered into the cytoplasm. Then laser induced DNA delivery is also reported. Ultrasonification, then macroinjection is your hypodermic syringe which we were talking about, where it can be directly injected into the cytoplasm or into the nucleus. Then microprojectile, which is your gene gun method, in which at a very high speed it can be delivered to the plant cells or the explant kept under an inert atmosphere with helium gas and the water vapor. Using the electrical charge under high voltage, the water droplets, high speed is created and it carries the extracellular DNA coated with the inert material, metals like tungsten or gold nanoparticles, which can be injected under high speed directly into the explant. I stop here, and then next class we will begin with the plant cell bioreactors. That is the last part of the course.